Hello everyone, welcome to another episode of discussion on how to make a presentation. Anytime we present in a lab, in a conference, we always want people to remember our story after the talk. Like if we have 5, 10 talks in a row, people hardly remember one or two talks and only the summary, not all the data someone has presented. So why that happens? Because not that the science is not good or the story is not good. It's the only difference is the story was not told to the audience in a way they can remember, or the presentation quality was not that good so that the audience could remember. So because of that, many times we lose the audience. Most of the time when a speaker is speaking in a podium, many people do not get connected with the talk, and then they get distracted with other things on the phone, or they simply do not connect and then do not understand or remember the talk. And why it happens is, one thing is the content, and the second thing is we do not connect with the audience, we do not consider what the audience need. So those are two things need to be remembered by the presenter: one, what the audience needs, and what are the material he or she is presenting. If both doesn't catch the eye of the audience, then you will lose the audience in the middle. So, in the today's episode, we'll be discussing what are the key things one need to remember, whether you are a student or anybody else who is presenting in public or to students. Please consider those points so that you can really connect with the audience and people remember your story. As scientists, how we always talk is, we start with introduction, background, then the rationale, then methodology, then results, data supporting our hypothesis, and then finally we tell the conclusion. But the problem is, what the audience listen is different. Uh, Imagine you are looking at TV news. They don't tell the whole story before telling the headline. They tell the headline first, what catch the eyeballs of the audience. Then they tell the whole story slowly. So, as scientists, also we should do the same thing. Otherwise, you will not have the audience listening to you. So, as scientists, we should start with the headline or the title page that catch the audience, and the people should be curious to know more what has been done in this project or in this story. Then the problem, why you are working on something, and then the methodology, then all the results, supporting data you can present, and then finally you give the future of what you are working on and where this story is going. And so with that, people will remember and they will think, okay, in future this is going to happen, so they might be interested in knowing more in the future. Okay. So today we'll be discussing how you make a PowerPoint PPT, and when you present, what all things you need to consider for the audience to remember your story. And when we prepare a talk, like most of the time, we present one talk everywhere, wherever we go. That's not a good idea, actually. So the talk should be tailored for the audience, and the title should give you a story that people can remember. And I remember this presentation is not a manuscript. So people do not need to see all the data, all the controls, all the experiments you have done. They just need to know what you have done. You can show all the data in a promotion presentation. If you are a student, you are going from JRF to SRF, or if it is a thesis defense, you need all the data to be presented. But if it is for a general audience, for the students or people who do not know your work, then you really do not need to show them all the data, all the PCR, all the Western blots you have done in your lab. That's not required for them to remember. And always don't prepare the talk to impress people. It should be for a story. You should be telling what people can remember. Never think that people will be impressed by the number of data, Western blots, PCR you are showing. They will not. Okay, they might be impressed, but they will not remember your story, because you will lose them in the middle if you try to show them lot of things which they do not understand. So the presentation has to be tailored for the audience whom you are presenting. As I've been repeating, it is all about the story you are telling to the audience. So for a scientific talk, if we see what we should start with, the field. Like our field had a magical word, then there was a problem, and we set out to address it, and then we developed a technology to solve the problem, and the tool now addressed all these problems, and we return to the field with new technology or the knowledge. But there are limitations, so we also should highlight on the limitation of the story we are working on, and then efforts are underway to address the limitations, and then happy end with the implications and the future, how this technology or new knowledge can be used. So that should be the last one, with the conclusions. And then finally, there should be a good thank you slide. Some people can bring this thank you slide to the beginning, because sometimes you are rushing through the slide and you will not have enough time to acknowledge people or the funders who have given funding and given their time and efforts to do the story, because you are not the only one who worked on the story. So the acknowledgement can be at the beginning, just to acknowledge everyone who has worked on this story. Let's take an example of a presentation. So how we need to make the slides and how we need to present to the audience. For a general public, like people who have no idea about what we are doing in the lab. So as I said, like the presentation need to be focused on the audience. And let's say this presentation is focusing on the researchers who are in the biotech field or life science researchers. So this presentation targets to researchers from scientists, students in master's or bachelor's degree, or university lecturers and professors who can understand what we are going to talk in this presentation. So let's start with the title page. So when we make any title page, so it should have the broad information about what you are speaking. So in today's presentation, we'll be discussing the development of a RNA isolation kit that was developed in our lab and then get commercialized by the RNA Biotech company, which is a spin-off from Institute of Life Sciences. So as you can see here, I didn't read the title, and you really do not need to read the title. You just say the background, or possibly if you want to highlight more, just tell them what people will be learning from this presentation, because the title is already there. They can always read it. You really do not need to read letter by letter here. Then in the second most important thing in any presentation is use dark letters against light background, or the opposite is also true. You can use light letters on dark background, but avoid red green combination. So sometimes people think, oh, my slide will look fancy if I make more colors, but it is not, because many people cannot read this red green combination and they cannot see it. Even if people can see it, it's uncomfortable for them to read. Okay. So, and remember, many people are colorblind. As you can see here, the middle one, most people can read it, but the left one and the right one, many people cannot read it. If you can read all three, that's good, but there are many, uh, especially male people who are affected with color blindness, they cannot see this. And same here in this one, a colorblind person cannot read any of this. So you need to consider every person in the row. You cannot make it very colorful for the normal people. You should also consider colorblind people because they cannot see the combinations. The second one is like the Maxure All RNA Isolation Kit developed by RNA Biotech Private Limited. If you see this slide, this shows a picture of the launch of the product. And what we learn from this slide is the font are in Helvetica. So that is the font that need to be used in, whether it is inside the slide or on the headers. That is the font you need to use. And here you cannot read RNA Biotech Private Limited very comfortably because it's in all caps. So you need to avoid all caps. So sometimes people think, oh, all caps is good, they will look nice, but it is not readable. So avoid all caps. It should be always in sentence case. Then the next slide, you come to the problem why you are working on developing this kit. So there were a lot of issues like the DNA contamination with the column based kits in the commercial kits available in the market, and these column based kits cannot isolate RNA. Those are less than 200 nucleotide. If you need smaller RNA or microRNA, you need a separate kit. And as the columns are limited, you are limited with the number of samples, so it is not scalable. And then the columns need higher volume for the illusion, and you need a centrifuge for all these steps for the isolation of RNA. So with all these issues, know, and there is a need for the development of a better kit. So in this slide, what we are learning here is you have clear text as less as possible and a bigger figure, no white space. The whole slide should be covered, and then large font size so that people can read it. Okay, so that is the second part of the talk, the problem why you are working on something. Then the opportunity, because we saw an opportunity ahead for developing DNA free total RNA isolation kit. And in this slide, what you see is a picture, no text. I could have written 2 3 lines or a paragraph on why we need to develop a kit, but I just put the opportunity I had. So pictures are better to connect with the audience, or even video. If you can put video, that is fantastic. You can put video because people connect with that, but do not have like a one paragraph write up on the slide, so that you will lose the audience. Okay. In this slide, what we say is we developed the affordable Maxure All RNA Isolation Kit, and it has many features like low volume RNA isolation to DNA free total RNA and microRNA isolation, and it can be scalable. in this slide, the slide is clear with the image, and you see there are small text and the bigger size text. So the small size text no one can read, if especially they are in the back rows. So you need to have any text on the slide that is not less than 18 font size for better readability. And the text, whether it is in the header or in the slides, body of the slide, it should be consistent. If you are using 28 font size for the header, keep it consistent throughout the presentation. Okay. Then in the next slide, what we see is the process. This is like supporting data, what we have developed and how it works. For how it works is, you have cell-lysate, then you filter it through a column, and then you get rid of the genomic DNA. Then the flow through is incubated with alcohol and beads, and then you get total RNA or the microRNA here. And in panel B, you can see you have total RNA, long RNA, and microRNA separated separately using the same kit depending on the need. So in this slide, what you basically learn is the slide should be clean and no more than two panels, and as less text as possible. And if there is a real need, uh, just put one or two lines of text, no more than that, and it should be big enough for people to read. Then the other thing you focus on is the supporting data again, the results and discussion part, like what this kit is doing. So as you can see here in panel A, we have used many other kits along with our in-house protocol for RNA isolation. And what we saw is in the PCR that works as good as the other commercial kits. In panel B, you see the CT values for mRNA, circRNA, and microRNAs, and they have as good as they have worked as good as the commercial kits. And even for the microRNA, our kit worked better. So in this slide, what we have seen is, explain one panel at a time. Don't put all panels at one go so that the audience is distracted. So, and they don't know which one you are talking about. So talk one panel at a time, then the second one comes. Even you can have three panels, you can, but they should not come at one go, one panel at a time, and then explain everything. If you are not explaining any of the data or any panel in the slide and saying, oh, these are the 10 things we have done, don't put them. Just put two things and say, we have done 10 different other ways, or we have tested them in five different cell lines and the results are the same. But don't put all five cell line data into one slide just showing you have tons of data, because people will not see what you have done. So restrict the panels to two to three maximum, not more than that, and explain everything. Then in the concluding slide, you just say what you are concluding from the whole story, what the takeaway message from the whole research you are doing. So in this case, what we say is, make sure all RNA isolation kit is one of the best kit in the market, and it can isolate RNA from different types of cells and tissues at a fraction of the cost. And it is developed by RNA Biotech Private Limited. So that is what the conclusion should be: clear, clean, only few lines on the concluding slide, not more than that. And remember to have the headers as the conclusion of each slide, so that people who cannot see what you have written in the text, or if they miss your talk by some means, they can always read the title and know what you are concluding from that slide, from the header itself. And if you read all the titles from the start to end, actually you should be able to tell the storyline. So let's see how our storyline went based on the slides I have presented. So that was the first slide, like Max Your All RNA Isolation Kit by RNA Biotech Private Limited. Then there are a lot of issues with the commercial column-based RNA isolation kits. There is a need to develop magnetic bead-based kit for DNA-free total RNA isolation. Then the next heading was we developed the affordable Maxior All RNA Isolation Kit, and the Maxior All RNA Isolation Kit can isolate total RNA as well as microRNA based on our data. And then Maxior All RNA Isolation Kit works as good as other commercial kits. And then finally concluding that Maxior All RNA Isolation Kit is one of the best kit in the market. So this is basically your conclusion. So if you collect all the conclusions or the headers from each slide, people should know what is your story. And this part people should remember. They may not remember all the data, but they should remember the conclusion of your story. And here the conclusion is Max Your All RNA Isolation Kit is one of the best kits in the market for RNA isolation. And finally, don't forget to put a slide for thank you. As I said, it can be in the beginning, it can be at the end. If you restrict yourself within the time schedule, put it at the end. Otherwise, if you doubt, you might shoot up the timing, you can take it to the beginning so that people uh, who actually worked should be acknowledged. And in this case, I acknowledge my lab, the company RNA Biotech who developed the kit, and the Institute of Life Science and the Bioincubator who support in the commercialization of this kit from development to the product. Okay. And finally, remember, giving a good talk has nothing to do with the amount of data you are presenting, number of Western blots, RT-PCR, facts, data. Nobody cares. What people care is the story you are telling them. If they do not remember or understand the story you are telling them, then it's a waste of time for everyone. So people should remember what you are working on, and the memory is short for everyone. So make it easy for everyone so that they remember your story. And hope this was useful, and you can make better presentation after seeing this. And hope this will help you for making a very good presentation that audience remember. Thank you.